This short video follows on from part 2. Here I will show the outcome of the transformation experiment. I will also show the sequencing data for the gene knockout. The first video covered PCR primer design for the gene knockout. In the second video, the PCR construct was confirmed and the transformation process explained. In this video, I will present new sequencing data, proving the PCR construct was successfully transformed. The data will show the before and after sequencing results, highlighting the gene knockout, as well as transformation of the construct into the correct genomic location. I will also show the effect of the kanamycin gene insertion. This is highlighted by a significant increase in resistance to kanamycin. The original, wild-type strain had a resistance of around 30 mg per liter. Following transformation, resistance to kanamycin increased to over 100 mg per liter. Here you can see the wild-type, as well as the transformed isolate. The transformant is growing on the top half of the plate. The wild type is on the bottom. This plate contains kanamycin at 100 mg per liter. This next part of the video will look at the sequence data. The following is a recap from video 1. There are 3 coding sequences predicted. To help distinguish them, these have been highlighted in different colors. We want to knock out the gene highlighted in yellow. It is worth noting, this gene has a ribosomal binding sequence and promoter. We will try and keep these. Looking at the DNA sequence again, the 2 flanking coding sequences can be seen with the gene of interest removed. The highlighted sequence in red illustrates the gene we want to insert within this region. For this example, the sequence is a kanamycin resistance gene. We will also design our primers to keep a promoter sequence for the kan gene. The promoter will be from our genomic sequence as highlighted earlier. The previous short clip highlighted what we set out to do. Following transformation, sequencing was carried out to confirm the PCR construct had incorporated into the genome, and to check it had incorporated in the right place, creating the knockout. Considering the circular bacterial genome, we want the kanamycin resistance gene to knock out a specific other gene. Should the kanamycin gene be at another genomic location, and expressed, this would not be a success. The wild-type and transformed isolates were sequenced using an Illumina platform. The sequence data was then uploaded to RAST. The annotations were downloaded in Excel for quick comparisons. In our original data, a flanking gene is shown in green. The gene we want to knock out in yellow. And a downstream flanking gene in blue. In the sequence data for outparent strain, with no gene knockout, the genes are identically arranged to those in our originally sequenced isolate. A side-by-side comparison of the 2 annotations confirms this. Regarding our isolate with the gene knockout, one of the flanking genes is shown in green. The kanamycin resistance gene is shown in red. The downstream flanking gene is shown in blue. While the knockout appears successful, from the annotations, it is necessary to check the sequencing data manually. This is the sequence data from the parent isolate. The flanking genes are shown in green and blue. The yellow region highlights the coding sequence for the gene under investigation. This is the sequence data for the knockout mutant. The coding sequence for one of the flanking genes is shown in green. The coding sequence for the kanamycin resistance gene is shown in red. The downstream flanking gene sequence is shown in blue. So far the knockout looks successful. Thank you for listening.